The ITAMs associated with the B cell and T cell receptors are phosphorylated by protein tyrosine kinases of the SRC family. Phosphorylation of the tyrosines in ITAMs serves as the first intracellular signal indicating that the lymphocyte has detected its specific antigen. In B cells, 3 protein tyrosine kinases of the SRC family, FYN, BLK, and LYN, are thought to be responsible for this. These SRC family kinases can associate with resting receptors through a low-affinity interaction with an unphosphorylated ITAM, to which they bind through a site in their amino-terminal domain. When the receptors cluster after antigen binding, the receptor-associated kinases phosphorylate and activate each other and are then thought to phosphorylate the tyrosine residues in the ITAMs of the cytoplasmic tails of immunoglobulin beta and immunoglobulin alpha. Phosphorylation of a single tyrosine in an ITAM allows the binding of an SRC family kinase through its SH2 domains. And kinases bound in this way are in turn activated to phosphorylate further ITAM tyrosine residues. The initial events in T cell receptor signaling are also implemented by 2 SRC family kinases, LCK, which is constitutively associated with the cytoplasmic domain of the co-receptor molecules CD4 and CD8, and FYN, which associates with the cytoplasmic domains of zeta and CD3 epsilon chains upon receptor clustering. The enzyme activity of the SRC family kinases is itself regulated by the phosphorylation status of the kinase domain in the carboxy terminal region, each of which has regulatory tyrosine residues. Phosphorylation of the tyrosine in the kinase domain is activatory, while phosphorylation of a tyrosine at the carboxy terminus is inhibitory. Even after being phosphorylated at the activating tyrosine, SRC family kinases can be kept inactive by a protein tyrosine kinase called CSK, C-terminal SRC kinase, which phosphorylates the inhibitory tyrosine. As CSK activity is constitutive in resting cells, the SRC proteins are generally inactive. An agent that counteracts the effects of CSK is the transmembrane protein tyrosine phosphatase, CD45, also known as leukocyte common antigen, which is required for receptor signaling in lymphocytes and other cells. CD45 can remove the phosphate from phosphotyrosines, especially from the inhibitory tyrosine residue of SRC family kinases. Thus, a balance between CSK, which prevents the activation of SRC family kinases, and CD45, which restores their potential for being activated, determines the signaling activity of SRC family kinases in response to receptor aggregation. This is one way in which the threshold for initiating receptor signaling is regulated in lymphocytes. Activated effector T lymphocytes and memory T lymphocytes express an isoform of CD45 that associates with the T cell receptor complex, thus facilitating signaling through the receptor. By contrast, naive T cells express a CD45 isoform that does not have this property. And that's the ITAMs associated with the B cell and T cell receptors are phosphorylated by protein tyrosine kinases of the SRC family. Have a great day.